Good morning. Good morning. Hey everyone, how's it going? Another week, moving through the semester here. Let's see. Let's pull this up. One thing I should mention is before I know people probably ask about it, I haven't been able to I haven't unlocked the first assignment yet. So some of you probably frustrated about that. Unlocked meaning like make it so that you guys can see the questions and the answers and what you got wrong and what was correct and stuff like that. It's because there were some late registry, like some students get registered late. So I'm still working with that issue, but it will be made available very soon. And then now the second assignment was due today. So the answers for the second assignment should be made available to you within 2 days, just so you know. So now that the late registration period is done, we shouldn't have that problem anymore. But I apologize, since I said last time that we were going to, since that was going to be available, right? But it will be soon, I promise. I'm sorry about that. The late registration thing kind of put a little stick in my spokes, as it were. All right, I'm going to share this. Let's see here. Okay, great. Make it big. Make it big. Come on, let's go. All righty. Hopefully you guys can see the PowerPoint slide. If you can't, let me know in the chat, or even feel free to speak out. There's snow blowing and like doing snow. It's been snowing here for days, but we don't have that much. But just so you know, there might be a little noise from people using snow blowers and stuff outside. I'm sorry about that too. Okay, let's get going. Yeah, so just a little reminder that the assignment number 2 is due this morning before 10, so right before class. If you missed the due date for this, just make sure you email me as soon as possible so that we can talk about that. Did someone have a question? No, okay. Yeah, so you make sure you email me right away if you missed the due date. All right, last time we covered all the bryophytes, right? We finished the mosses, then we went through the liverworts and the hornworts. So the bryophytes are done. And then you had them in lab. Hopefully lab went okay. I'll be meeting with the lab TA today to kind of troubleshoot and think about ways to improve lab. So just so you know that as well. Now we're gonna leave the vascular plant, the non-vascular plants behind. We're gonna start talking about vascular plants today. In particular, we're gonna talk about parts of the plant body in the vascular plants and like how those parts evolved from non-vascular plants. We'll also talk about some tissues in growth, not too much about tissues in growth, but some. So there's going to be a little bit of tediousness today about that. And then some of the phyla of the early vascular plants. So I always like to get oriented, right? Like, where are we? So we've covered the non-vascular plants, and now we're going to move into the bulk of the plant kingdom, which is composed of the vascular plants. And although we won't be talking about them in detail today, we're going to start by talking about the spore plants, which are the fern, the lycopods, and the horsetails. So we have to talk a little bit about how plants went from being non-vascular plants to being vascular plants. Like, why even do that? If being non-vascular works, then what pressure is there to make vasculature? Well, some important things to consider about non-vascular plants is that we know from the name they don't have vascular tissue, right? The other thing we know is that they have the gametophyte generation dominant. right? The mosses, the liverworts, and the hornworts all have the gametophyte generation dominant. And we know in the non-vascular plants that the sporophyte plant is reduced and dependent on the gametophyte. Reduced meaning small, right? So the sporophyte plant compared to the gametophyte plant in the non-vascular plants is very small. And the sporophyte plant, in addition to being small, also always has to be attached to the gametophyte plant in the non-vascular plants. The reason the sporophyte plant always has to be attached to the gametophyte plant is because most of the time, except in the hornworts, most of the time the sporophyte plant is non-photosynthetic, right? We did see a little bit in the hornworts, there was some sporophyte, the sporophyte plant was photosynthetic, but in general, the sporophyte plant is non-photosynthetic in the non-vascular plants and therefore can't make food. So it has to be attached to the gametophyte plant in order to live and get food and water. Water. Oh, thank you, sporophyte plant. No, I'm just kidding. Okay, so those are some important things to consider about with the non-vascular plants, right? Well, so some of these things are great, right? Like, so the pros of being a non-vascular plant is you're very simple in structure. So you don't really need that many resources. But there's some definite cons to being non-vascular plants. Number one, you're simple and you don't need that much resources, but you're also pretty inefficient, right? For example, when we're talking about the mosses, we're talking about how things move through the mosses, right? And it was through osmosis and diffusion. Osmosis and diffusion work, but they're not very efficient processes and they're slow. Another con to being a non-vascular plant is you can't get that big. I mean, like you see every time we talk about the mosses, liverworts, and hornworts, right? They're little green blobs, basically, little tiny green blobs. You'll never be walking through the forest and see like a moss towering over you, right? No, because they're so simple that they have also very small sizes. They release spores right into nature, right into the environment. So the spores are not protected. So that's another con. And in order for them to have fertilization occur, they have to have water, right? Because the sperms swim. And if you don't have water, it's pretty hard to swim. So, so you need water to be present if you're going to have fertilization occur in the non-vascular plants. And if water's not there, you can't have fertilization occur. So that's a con. And last but not least, each sporophyte plant only has one sporangium. So remember, in the non-vascular plants, we called that sporangium a capsule a lot of times, right? You can call it a capsule, but each sporophyte plant is only capable of making one sporangium. And that's not great, you know, only one, that's not great. So these are the little pros and cons of being a non-vascular plant. Now vascular plants. So we had that design of like, okay, I'm a non-vascular plant and I can do these certain things, but I'm limited in these certain ways. And now we're going to talk about the vascular plants. So as the name might imply, right, vascular plants have vascular tissue, right? Because they are vascular, vascular plants. The other big, so these important characteristics, you should compare them to one another, right? Like these, so non-vascular plants, no vascular tissue. Vascular plants, vascular tissue, right? Non-vascular plants, free living gametophyte. Vascular plants, free living sporophyte. So now in the vascular plants, the sporophyte generation is dominant. The sporophyte generation is dominant and free living meaning no longer attached to and dependent on the gametophyte plant. Free living, and it's the dominant generation. And then now we have a reduced gametophyte, whereas in the non-vascular plants, we had a reduced sporophyte, right? Got a question. Did non-vascular plants come first? In evolutionary time, they did. Yes. Yep. And based on DNA analysis and also the fossil record, and but yes. So the non-vascular plants. Remember that plants started in water, right? They started in aquatic environments to begin with, and then ultimately made the transition from a life aquatic, like in a life in water, to life on land. And those first plants that made it on land were non-vascular. And vascular plants show up much later in evolutionary geological time. Yes, good question. Okay, so yeah, comparing the important characteristics of the vascular and non-vascular plants. So they're kind of opposite, right? They're kind of opposite to one another. All right. So why vascular? Why? If the non-vascular thing worked, what pressure is there to do vascular? And we covered some of the cons in the previous slides. So now we can talk about the pros and cons of being vascular. So if you're a vascular plant, number one, you can get much bigger than if you were non-vascular, right? So like a tree, for example. You will walk through a forest and see a tree towering over you, and a tree is an example of a vascular plant, right? So you can get much bigger. Number two is that now the spores are going to be actually protected from the elements, so that's a plus. And you no longer need water for the sperm to get to the egg, which is a big one, because if that's a very limiting thing. If you need water for sperm to swim, it really limits when you can have fertilization occur. And then the vascular plants no longer rely on water for that process. And multiple sporangia per sporophyte. So one sporophyte plant now is going to be able to make lots and lots of sporangia, and hence lots and lots of spores. But the cons are that they're much more complex and therefore require more resources. So just so you know, these two slides here, this slide, right, and this slide, these are good slides to make a note of, because these are jammed packed with information. But notice that the one thing I want you to notice is not only are the important characteristics opposite, but also the pros and cons are opposite, right? The pros and cons are opposite for these two things. So there you go. That helps you compartmentalize it and maybe remember. All right, so now we'll talk a little bit about these early vascular plants. So here, this picture kind of shows an idealized version of what like a very early forest might have looked like, right? A very early forest might have looked like. So that's where we're putting our minds now, that we're like in this very early forest. And this here shows you there's different geological eras on the left, and showing you when things started to show up. So take a peek here. That question before about the non-vascular plants were first. Yes, so look, oops, come on, hold on. Yeah, let me annotate. Let's pick, what color should we use? I guess dark blue, that's good. So check it out. We have here, so here's the bryophytes, right? We had algae was in the water. And then here's the bryophytes. When do the bryophytes show up? Like around this time, give or take. And then we get to these so those are non-vascular plants. Then we get to these vascular plants, right? Ferns, horsetails, and lycopods. And those show up right around here. So this is in millions of years ago, right? So you're like around, less than 200 million years ago is when these guys show up, and more than 200 million years ago, even longer, is when the bryophytes showed up. Oops. Oh, I always forget that I do that. It looks like algae came after bryophytes. Actually, sorry, I just realized I made a mistake on this. Sorry, I was circling, good catch, Catherine. I was circling the drawings. I apparently need more coffee. You can't see all right, so the pictures are not when they showed up. I thought that was wrong because the mosses are like 400 million years old. I'm not a morning person. Look at this down here. Look, there's an arrow down here going later in time. You see that? Yeah, so this algae is connected to this line. I was wondering why that line was there. It's pointing to this arrow to go back, right, into even further in evolutionary time, more millions of years ago. Bryophytes, see, this slide is focusing on the early vascular plants. So the bryophytes and the algae didn't even make it onto this. Look, bryophytes can't see, but they're also around here 400 million years ago. Look, early vascular plants appear about, there we go, 410 million years ago. So that's these guys. My bad. Sorry about that. I was circling the picture. This is why I should look at these slides before I do this lecture in the morning. Yes, good catch. Thank you. Yeah, so sorry about that. Yeah, early vascular plants about 400 million years ago. Mosses and algae way before that. Way before that. Obviously, if I don't know this by heart, I don't need you to memorize millions of years ago. But the point is, old. Early vascular plants, super old in time. Okay. Oh, see, I should have just gone forward. I circle it. I circle it for you guys. Well, at least now when you guys are looking at the slides later, this will be here and you won't get confused about what I'm looking at. Okay, so early vascular plants show up around 400 million years ago. Why do we care? Well, look at this. So these are animals. Oh, thank you. So that previous slide was showing you plants, right? Plants. Now, animals. So early vascular plants show up around 400 million years ago. Okay, well, what else showed up around 400 million years ago? And I think I probably color on it. Yes. So here you go. First land plants, right? So that's your bryophytes, or actually the ancestors of bryophytes, showing up for around 400 million years ago. And at that same time, look here, then we get to the first vascular plant showing up around that a little bit later, right? About, 50 million years later. But look at what shows up when those first vascular plants show up. These things right here. Here, sorry, that was a confusing way to show you that. Here, you'll use yellow. Look, tree-like ferns, right? That's telling you vascular plants on this slide. And look at all this. So before that, we have like these type of animals, fishes, right? Corals, things in the ocean. Then we get first, land plants are already on land. Now we're starting to get animals leaving the water and coming onto land too. And then next thing you know, oh man, insects, reptiles, etc. So when plants transition to really making their life on land a permanent thing by making these vascular structures, likewise, we see a switch in what types of animals are occurring, because the plants are giving the animals resources on land, so there's an incentive for animals to also transition to land. So as plants dominate the land, animals also move on to land. That's kind of important of a thing. Okay. Oh boy. All right. Clunked my way through those slides. All right. So we're in the vascular plants now. We know now the Sporophyte generation is the dominant generation. So no more green fluffy. green fluffy as the dominant generation? No. Now sporophyte generation is dominant. And for those early vascular plants, when they're first kind of getting established on land, things that they have are a root system. So because they're vascular now, remember in the non-vascular plants, we would say, oh, well, they have like stem-like parts and leaf-like parts and all that. That's because those didn't have vasculature. Now that we're talking about vascular plants, we have these real things. So we have root system, a shoot system, which is your stems and your leaves, and then the interior vasculature, which we'll talk a little bit about. And these plants make spores, not seeds. So we're still in the spore plants, not in the seed plants yet. And I will say, obviously, these plants are just starting to become vascular, right? These early vascular plants. So we do say it has like primitive roots, primitive stems and leaves, and primitive vascular. Don't worry about stele yet. Vasculature. And that just means that they're kind of like transitioning on their way to being what we now have in modern times in terms of structures. Okay. So let's look at this plant. This is, what do we know from the fossil record? Well, so it's not like you go from like like a non-vascular plant to like whammo vascular plant, right? Like there's transitions in time, right, as plants adapt to this life on land. And so because of that, in the fossil record, we actually can see some sort of intermediate plants where they're kind of still, they're not quite a non-vascular plant and they're not quite a vascular plant yet. They're kind of in the middle. So this is an example of that type of a plant, this intermediate type plant. That's Aglaophyton. And this is an extinct plant that occurred around 400 million years ago as plants were transitioning to becoming vascular. And notice, so this plant doesn't have any leaves. This plant doesn't have any roots. So that's similar to a non-vascular plant. It does have a photosynthetic stem. And it used rhizoids, which you could actually see, not super great in this, but these little nubs down here, they have little hairs coming out of them, and those are the rhizoids to attach it to this, whatever it's growing on. So that's similar to the non-vascular plants, right? Rhizoids. The stem grew horizontally, meaning along the ground. It's just a description. And had sporangia located at the tips, right? Or the stem axes. Sporangia located at the tips. So that's also similar to your non-vascular plants in that, like, think about the mosses, right? Where did the sporangia occur? On the top, right? So that's similar to the non-vascular plants. And this intermediate plant had this central stand made up of leptoids and hydroids, which we also saw in the non-vascular plants. The difference is, though, and I guess I should make this more clear on this slide because I'm just now realizing didn't make this clear. Think about it. So here we're saying no leaves, no roots, photosynthetic stem, rhizoids, sporangia on the top, and leptoids and hydroids. These sort of things here, the no leaves, no roots, so photosynthetic stem and rhizoids, those are things that are similar to non-vascular plants, gametophyte generation. But this here that we're looking at, this Aglaophyton, that is a sporophyte. That is a sporophyte with a photosynthetic ability. So the ability to live independent of the gametophyte plant. And the sporophyte plant now, sporophyte plant now is what has these leptoids and the hydroids. Remember, when we talked about leptoids and hydroids before, it was in the non-vascular plants gametophyte generation, which was dominant in those. Sorry. Like I said, they're doing snow work outside. Okay, so that was Aglaophyton. A little bit later, probably a contemporary, but a little bit more, a little less intermediate, I guess we can say, is this earliest vascular plant. So this was not technically a vascular plant. This was an intermediate plant making the transition from a non-vascular existence to a vascular existence. This, though, is a vascular plant. So this is the earliest vascular plant that we know of so far, that we know of. It's something called Cooksonia. Cooksonia. And it's in this phylum, Rhiniophyta. You know, don't worry about that. Just know that this that we're looking at right now is the earliest vascular plant. That's what you need to know. They are extinct, obviously, and oops, still no leaves, still no roots, still has a photosynthetic stem, still use rhizoids. So all three of these things are also characteristics that the intermediate plant had that we just looked at. Still has a horizontal stem, still has sporangia located on the stem axes. But now, finally, we finally are starting to see this vascular tissue develop, right? So a primitive type of xylem and a primitive type of phloem, but it still has xylem and phloem. This picture in the middle down here is actually a picture of a fossil of Cooksonia, and it's showing you the xylem and the phloem tissue as versus the leptoids and hydroids that intermediate one had, right? Look at these here. Leptoids and hydroids in the intermediate. Real xylem, real phloem, although a little bit primitive in this earliest vascular plant, Cooksonia. This is kind of so this is a relative of that Cooksonia, another thing in the phylum Rhiniophyta. This is Rhinia. And all I'm showing you here is like what the fossil record shows us about how these plants changed and became more complex over time. This is a picture of a fossil of sporangia from this extinct plant. And this is actually fossilized spores as well. That's kind of cool. Okay, so we had our intermediate plant, we had our earliest vascular plants, and now our slightly later vascular plant. So this is something called Saudonia. It's also an extinct plant. And let's take a look here and see how Saudonia is a little different than the Rhiniophyta we just looked at, right? Rhiniophyta, we just looked at this. So let's look at how this is a little different. So first, it has the exact same characteristics as this. So it has no leaves, has a stem that is photosynthetic, etc. Has the exact same characteristics. The only difference is now the sporangia actually grow laterally, meaning along the sides of the plant, not at the tips. Why is that? Why could that be advantageous? Well, look at this. Let's go back. If you could only grow sporangia from the tips, right? Stem axes. That's only up here. And you only have so many tips, right? You know, there's only so many tips. It's kind of like you're missing, there's this whole rest of the plant that is being underused, right? If you only have them growing at the tips. So that's why later on, with these slightly later vascular plants, now sporangia are growing all over the sides of the plant. That's great because now you only have, like if this is you only have one tip, right? But you have all this surface area along the sides. So if you can cover that surface area with sporangia, that means that each sporophyte plant is going to be capable of making many more sporangia than if they were only making them at the tips. So lateral sporangia, big deal. No, it is like a real, it is a big deal. And then the other thing, so the other way it's different than the Rhiniophyta we saw on the previous slide is that it has this lateral sporangia, but it also has xylem and phloem that look like the xylem and phloem that you can see in plants living today. So modern xylem and phloem, as versus that primitive xylem and phloem in the earliest vascular plants. Oh yeah, and then you might be seeing these little spiky things. Maybe you're thinking those are where sporangia were and maybe they fall off. No. These little spiky things, or I said here, spine-like things, are called enations. And we'll talk more about enations later on. But enations are the what eventually led to leaves, leaves. We have a question here. Does growing lateral also protect the spores from the elements? No, not really, because here, I'll get rid of these drawings. You know, these guys here, first of all, keep in mind, these things are not spores. These things are the sporangia, right? The spores are inside of those. Spores are inside of those, and eventually the spores are going to get released out into the environment in these earliest of vascular plants. Oops. So keep that in mind here too. Even though these are at the tip, each of these things, here's one, are sporangium, right? Or plural sporangia. We have all three, and the spores are inside. So tips or on the side, sporangia growing at the tips, sporangia growing on the side of the plant, kind of no advantage in terms of protection. It's more that you can just have a lot more sporangia if they're growing on the sides of a plant body instead of only at the tips. Okay, and so enations. We'll talk about enations later on. Maybe today, I'm not sure. Okay. Just looking at the time. All right. So even though these that we just looked at, these plants that we just looked at are extinct, no longer around, there are still early vascular plants that live today that are still alive and not extinct. So if something is still alive today, you can call it extant, believe it or not. That's how, oh, yellow doesn't work really well in there. Extant means not extinct. Extant means still here. Let's go to red, I guess. Yes. Extant. So there's still, boy, one day I'll get the hang of this annotating. Not today though. Yeah, so we're going to talk about some of these right now. I got a question in the chat box. So the intermediate plant had more than one sporangium per sporophyte? Yes. The intermediate plant had more than one sporangium per sporophyte. Let's go back real quick. Yeah, I'll just show you here, Brittany, hold on. Oops. Here. So for all those vascular plant, any vascular plant that we talk about is going to have these things. So multiple sporangia per sporophyte plant. Even the intermediate one. Oh boy. Here we go. Close your eyes so you don't get dizzy here. Okay. So these are examples of early vascular plants that are still alive today. And these are pictures of them. What are we looking at here? Well, the ones we're going to end up talking about in the class, right? On the top, we have the lycopods. On the top and the middle. To the right of that, we have the horsetails. Oh, and and so there's lycopods and horsetails all over this slide, right? Top and middle, I guess left side and middle lycopods. Right side is horsetails. They still look pretty primitive, right? They don't look like a tree. The lateral spores plant are extant, correct? So this plant, which is Saudonia ornata, is extinct, no longer around. So no, so this plant is extinct. All of these plants that I just showed you, see, because this, look here. What do we know from the fossil record? Fossil record. So all of these plants on these slides that say the fossil record thing are extinct plants. So the Saudonia, the Kiksonia, the Rhynia, and the Saudonia, those are all extinct. Cool. All right, let's start talking about these a little bit. So, oh, right, tissues and cells. That's okay. We'll talk about them a little bit. Before we talk about these plants, we have to talk a little bit about their internal anatomy. So, if you're a vascular plant, you have certain tissues. We have already covered two of those tissues, xylem and phloem, right? Because you can't be a vascular plant without having vasculature, and the plants, that's xylem and phloem. But there's other tissues as well, and all the tissues inside of plants are organized into tissue systems. Tissue systems. And in the plants, there are 3 tissue systems. In the vascular plants, there are 3 tissue systems that occur in the sporophytes of the vascular plants. Remember, the sporophyte's dominant now. These tissue systems are the dermal tissue system, or skin, like the outside stuff. Ground tissue system, which is the tissues that are below the skin and the outside stuff. And then last but not least, the vascular tissue system. So these are the three tissue systems of sporophytes in the vascular plants. And I'm only showing you this thing now to kind of get you thinking about this. We're going to talk in subsequent classes, we're going to talk a lot more about these tissue systems and stuff. But the main thing I want to show you in this is that, so you have 3 tissue systems, but they all arise from the same type of growth. They all have all 3 tissue systems originate from the same type of cells. You could think of it almost like a stem cell, right? Like a meristem is kind of like a stem cell. But once things occur, chemical signaling occurs that we don't need to know for this class, and they are destined to be a type of a cell, they can become these 3 types of cells that ultimately become parts of these 3 tissue systems, right? So we have 3 tissue systems. In the dermal tissue system, you have the epidermis, the ground tissues. There's a whole bunch of different ones, some of which you'll cover in lab. And then for the vascular, so this is dermal tissue system, ground tissue system, and vascular tissue system, which is your primary xylem and primary phloem. Okay. I don't want to get too hung up on this today. I think we talked about it a little bit here. The Zoom is, okay. So what are the parts? So hold on, I'm gonna sneeze here. I thought I was gonna, I hate when that happens. Oh man, I thought I was gonna sneeze. All right. So here we talked, there's 3 tissue systems. Now, what do those tissue systems occur in? They occur in parts of the plant body, right? Or the organs of the vascular plant. What are these organs? Well, you have roots, right? Which serve to not only hold the plant to the ground or whatever it's growing on, but also to absorb water and minerals. We have a shoot system, which is your stems and your leaves. This is the primary parts of the plant that make food, right? They're green and they do photosynthesis. So the leaves are the main photosynthetic organs, and the stems are going to help elevate those leaves up so that they can be towards the sun. And then the vascular tissue system, which is your xylem and your phloem, and that serves to conduct water. The water is conducted from the roots to the leaves via the xylem, and the food made from photosynthesis is going to be conducted from the leaves down to the roots and other parts of the plant. And notice this: conducts water, conducts products of photosynthesis, aka food. I can't draw straight lines to save my life. Yes, so conduct. Why conduct? Because now we're more efficient. We're more efficient than we were in the non-vascular plants, right? These things make us more efficient. Okay. And since we are talking about a sporophyte plant, don't forget the sporangia, right? It's in the name. So that's also an important organ. And we know what the sporangia do already. All right, let's see where we are with this. Do I want to? Yeah, all right, we can do it. Yay, we can do it. We can do this. All right, so 3 tissue systems, right? Dermal, ground, vascular. Where are those? They're in all parts of the plant. So dermal tissue system, remember dermal, one way you can remember the thing that goes inside of a dermal tissue system is the epidermis, right? Dermal tissue system, epidermis. So if you look here, leaf, stem, and root, right? Epidermis. So here's a leaf at the top in A, and it has an epidermis, right? So that's that. Here in B, you got a stem cross section. Epidermis is on the outside. That's better. And then in the root down here, the epidermis is the layer that covers the outside of that root. It's a root cross section. Okay. Next, we have a ground tissue system. Change colors to keep things organized. The ground tissue system in the leaf, so here we are, a leaf, is called mesophyll. So it's all of this stuff. Oh, I should have done it green. That's okay. So all this stuff is a mesophyll. That's the ground tissue in the leaf. In the stem and the root, the ground tissue, your stem, is called cortex. And it's like all this stuff here. And in the root, same thing. All this stuff here. Whoa. So these are the parts of these tissue systems. And then last but not least, in the vascular system, the parts are called xylem and phloem, and doesn't matter where you are in the plant. So I'll use red. Here it is in the leaf, xylem and phloem. Here it is in the stem, xylem and phloem. Here it is in the root, xylem and phloem. Okay. All right. Right. Extant early vascular plants have perfect order. Yes. So this is kind of what I was going at before. in talking about these like kind of transition, we saw it in the fossil record in that some of the things that we see in the fossil record, they're kind of intermediate and maybe like, or once they do have xylem and phloem, the xylem and phloem is kind of what we say primitive. Well, the plants that are still alive today that we consider these earlier vascular plants, early vascular plants, like lycopods, horsetails, and ferns, are they're still not a tree, right? Like they still have some things that make them a little bit more, I guess, primitive than other vascular plants that evolved later, in particular the seed plants. So this slide is like a fancy way of saying the spore shedding plants that are alive today, that are vascular, are still a little bit more primitive than like the seed shedding plants. That's kind of what this slide is saying. This is kind of a silly slide. I want to get rid of this slide. But let's at least look a little bit about this. So just keep in mind when it says early extant vascular plants, that's talking about the lycopods, horsetails, and ferns. Right. Primitive, right? A little bit more primitive. So they do have roots, but they're a little primitive. They do have a shoot, but it's a little bit more primitive. The xylem and phloem organization is kind of like simple and primitive. And then how is this? Oh, right. Yeah, it's like okay, yeah. Yes. So think simple. The lycopods, horsetails, and ferns, which are still around today, and they are the examples of the early vascular plants that are still alive today, they have these characteristics. Later, when we're talking about more complex plants later on, they'll be more complex in these ways. And the only reason I point this out here, the earliest vascular plants are homosporous, or they only have one spore type. That's similar to the non-vascular plants, right? Like when we talked about the mosses, the liverworts, and hornworts, we didn't have different types of spores, right? There was only one spore type. And these earliest vascular plants also only have one type of spore. Later vascular plants have two types of spores, but don't worry about that right now. Plenty of time to talk about that later. All right, so let's look at some of these more primitive organs that exist on the plants that are around today. So here's, what do I mean by primitive roots? They're very simple roots. Look at these, simple. There's the roots right there. They're simple. They actually come out of the stem. So remember horizontal stem, said that on the previous slide. Horizontal stem is growing along the ground. When wherever the stem touches the ground, that's where roots will come out. Don't worry about that. And they have the tissues that we already covered on the previous slide, right? Epidermis, which is the outside part, cortex, which is the ground tissue, and then stele. Stele. So I tried to get away from having to talk about stele, but it still shows up. So we got to talk about stele. What is the deal with the stele? What is the stele? What is the stele? Well, I got to get rid of that thing first. Clear all drawings. Yes. Stele is a fancy way to describe how the xylem and phloem are arranged. So xylem, phloem, they're going to be organized differently depending on which plant you're talking about. And we name that organization is a stele. I know this is going to probably seem a little confusing right now, but we will cover stele a bunch of times later. I'm just getting you into it now. And these are the 4 types. Don't worry about this right now. I'm just showing you, don't worry about any of these words on here. I'm just showing you that there are 4 types of ways that xylem and phloem can be organized inside of a plant. There's 4 types of ways. And in the plants that we're going to talk about today, but mostly in the next class, have their xylem and phloem tissue arranged in what's called a protostele. A protostele. So that's all you need to know from this slide is that xylem and phloem can be organized in different ways. The way that we name that is called stele. There are 4 types of steles, and the earliest plants that are vascular have what's called a protostele. And I don't care about this. Protostele. So, right. Remember we looked at those plants from the fossil record a few slides back, right? This was the one that had the lateral sporangia, right? We said that this extinct plant had what we call modern xylem and phloem, right? Like the same type of organization that we see in modern living plants. And so what we mean by that is that this plant that we saw in the previous slides, right, had a protostele. So that is like how the plants nowadays have it arranged. And so that's what we mean by it had modern. Okay, I think we're almost done. Almost done. Hang in there. So that was, we talked about roots, we talked about the stele. Shoot system, which is your stems and your leaves. Remember, the stems here are going to grow horizontally. When you could call that a rhizome. Don't worry about that too much right now. We'll talk about that next time. Anytime you see this word in my slides, I think it was on a previous one too. Anytime you see this, all I'm saying is that the way that it grows is that grow split, like we saw in the liverworts. So here, grow split, right? Grow split. Look, grow split. So anytime you see this word dichotomize, that's all I'm talking about is this grow split idea, which is just a simple pattern of growth, right? Grow split is a simple pattern of growth. And we already know that this consists of these tissues that we covered in the previous slide of the stem, shoot system rather. Here is that protostele again. So all you should know about the protostele is that it is, first of all, it's a way that the xylem and phloem is organized. It's solid, meaning there's no hole in the middle. It's a solid structure. And it has xylem tissue in the middle. I guess I should change colors. Use pink. This here over here is just a close-up of what's here. It's just a close-up. It's not 2 different things. Xylem in the middle, or rather, that was not a good way to draw that. Xylem here. The xylem is the bigger stuff. Xylem that I'm drawing here in pink. And it's surrounded by, I guess let's use green, surrounded by phloem. It's not super great color, but anyways, here, phloem, phloem, phloem. And the point is, it's solid. There's no hole in the middle. Later on, we'll see ones that kind of look like with like a hole in the middle, but not today. These are solid. And this occurs in the stem and the roots. So that's protostele. Let's look here at the leaves. Remember the leaves I said are kind of more simple than later plants. So simple in this case means that they only have a single vein and they're small. and we call them a microphyll. Phyll means leaf, micro means small. Oh yeah, see? In the terms here, though, micro, it does mean small, but it also refers, so here microphyll means a simple small leaf that has a single vein. Later on in the course, micro is going to refer to something different. But for now, just worry about this microphyll, simple leaf with a single vein. Here's a cross section through a microphyll. And what does it have? It has an epidermis, so it's going to have your 3 tissue systems, right? It's going to have a dermal system, ground system, and vascular system, tissue systems. We have here epidermis on the outside. Stomatal apparatus, we talked about that during the hornworts, right? There's one right there, which is that area of gas exchange that's regulated by special cells. The ground tissue system, which is composed in the leaf of the mesophyll, that's all this area in the middle. And then your vascular tissue system, which is composed of the xylem and phloem in the middle. Remember, it's a microphyll, right? Microphyll. So each leaf only has 1 vein, right? Single vein. Microphyll. Small leaf with 1 vein. Okay. Oh yeah, we are going to talk a little bit. I think this is where we end, is with this Enation theory. So remember in the previous slide when we saw the extinct plant that had the lateral sporangia, and I said those little spiky doodads were the Enations, and I said on that slide that the Enations are thought to be the precursor of leaves, or if, in other words, plants first had these little weird Enation things that over evolutionary time ultimately became these simple leaves or microphylls. So you have Enations, and then you have evolutionary time and pressures, and then ultimately microphylls. Here's what they think happened based on the fossil record. Here's what we think happened. So basically you would have had these, here's a simple stem, right? The darker stuff in the middle is the vascular tissue, right? Eventually you end up with these little projections, right? That are those Enations. But notice this Enation here, there's no, this one here. There's no dark stuff in the middle. There's no vasculature in this Enation. It's just a little spike thing, but there's no xylem and phloem inside. But eventually that vascular tissue started to migrate into that Enation, such that eventually, once that does occur, you end up with what's called a microphyll. So see, the dark color in the middle is showing that's where the vasculature is now. So based on the fossil record, this is what we think happened. That's, so you can have it later for your slides. And then I think we are pretty much done here. So microphyll. Microphyll, remember, small leaf with a single vein. Some microphylls are able to have spores on them, right? In the bryophytes, we had sporangia located on like the tips, right, of the plant. But now in these earliest of vascular plants that are still around today, the sporangia sometimes are just on a leaf, And in the first case of this, it's sporangia that are on microphylls. And if a microphyll has a sporangium on it, we call it a sporophyll. So a phyll or a leaf that bears spores. So some microphylls have sporangia, some microphylls don't. If it doesn't have it, we just call it microphyll. If it does have it, we call it sporophyll. Yeah, that's what I just said. Oh yeah, sometimes the sporophylls look exactly like the microphylls that don't have spores on them. But sometimes the sporophylls look a lot different than the microphylls that have no spores on them. And we'll see pictures of this. When they look different, when they look morphologically different, the sporophylls will be smaller than the microphylls. They'll be not green, and they will be organized in something called a strobolus. There's a lot of new terms today. Don't get scared. We will have a lot of repetition in these terms going forward. Sporophyll, we're going to say 1,000 times. Strobolus, we're going to say 1,000 times. Microphyll, we'll say maybe a couple hundred times. So don't worry too much about this. You're just getting used to it today. Okay, this is an example of when the sporophylls look identical to the leaves that don't have spores on them. So I'm going to go forward because I probably draw on it. Let's see, because I tend to do that. No, I don't. Here. Okay. See these green leaves? Here's one, right? Here's one. All these spiky things are leaves. These little things on the base of those leaves are the sporangia. But the point is, look at the leaves. The leaves just look like green, normal leaves. They don't look different just because they have a sporangia on them. They just look normal and green. Oops. But sometimes they don't look like that. Sometimes they look completely different in that look. So here, this is an example of that. On the bottom, you have just the green normal leaves. These are the microphylls that don't have sporangia on them. But up here, in these things, those are actually composed of leaves. So like in here, these are all little tiny leaves that are on this thing. But those leaves are a lot smaller. They're not green, even though they kind of look green on this slide, but trust me, they're not green. And they're organized into a thing called a strobolus. So this whole thing here is one strobolus. Again, don't roll your eyes and be mad at me for saying scary words today. We will have a lot of repetition in these terms. But point is, sometimes the leaves that bear the spores look like the leaves that don't bear spores, and sometimes they look different. We'll see this as we learn about these plants. This is just a close-up to show you what it looks like. So this is a close-up. So here, this is what I was saying before. The microphyll, small leaf with a single vein. This microphyll does not bear sporangium, so we just call it microphyll. However, down here we have microphylls that do have sporangiums on them, and so we call those microphylls sporophyll. Sporangium is still sporangium, so it's still going to make spores. Here is the leaf. This is the microphyll that has a sporangia on it, aka sporophyll. Here is that sporangium on that leaf, and the spores are inside. Okay. Same thing, just a better microscope slide. Same thing, better microscope slide. Okay. These are what we're going to cover over the next several classes. The different phyla that are still living today, but are examples of these early vascular plants. Lycopods, horsetails, and ferns. In the next class, we'll start talking about the lycopods. So today we really focused on the transition from a life as a non-vascular plant to a life as a vascular plant. And we talked about parts of the plant body, particularly the sporophyte plant body in the vascular plants. Next time, now that we know all of this information, we can start talking about the actual players. So if anyone has any questions, I'm happy to answer them here. If you came in late, just note that I said at the beginning that I will be making the answers available to assignment 1 very soon. There's just been a delay in that because of late registration things that I'm still dealing with. Assignment 2 was due before this class began. If you didn't turn it in on time, please email me immediately so that we can set that up. And if anyone has any questions, I'm happy to answer them here, or you can feel free to email me as well. Oops. Have a good day, guys. Sorry if this class was a little boring. Sorry. Have a good one. I hope you guys have a great rest of your classes.
Have a good day.
You too. Oh, Mohammed, do you have a question?
Yes.
Okay, what's going on?
So actually, I took the quiz, but I was asking if the grade is that the final grade, because there was some questions having like, complete the blank.
Sure, good, really good question. So, Blackboard, so when you submit an assignment on Blackboard, Blackboard will try to grade it, but sometimes it's not perfect. And so I actually will open every single student's assignment and grade it manually. So the grade that first shows up might not be the grade that ends up being the grade. For example, in the first assignment, like sometimes I have you like fill in the blank and you might fill the blank in different ways, and
Yes.
your response will still be correct, but Blackboard might not think it is correct. So I go in and I override any of those errors that Blackboard makes.
Okay, thank you so much.
No problem. Have a great day.
You too.
Any other questions? Oh, thanks, Brittany.
Yeah, I have a question, Professor.
Yeah, Andrew, what's up?
So yesterday I was reading this thing that lemons were actually like a hybrid fruit produced by crossing, like I think it was bitter orange and citron, something like that.
So I'm not familiar with that. I will say that all citrus fruit is the result of intense breeding over a very long time. Like I don't want to say 1,000 years, but maybe.
No, so my question is, because I remember in one of the other biology classes I had taken before this one, the professors were explaining how if a cross between two species, if a cross between two individuals can produce, what's the word I'm looking for? Like hybrid.
Oh, like a hybrid?
Yeah. If they could produce offspring that could reproduce, I guess is the way I'm saying it, then they're technically the same species.
Yes, I see what you're getting at. Right. So in biology, we have this thing called the biological species concept, right? And it's how we say whether or not something is a species or not. And there's different ways to define whether or not something is a species, right? One way, as you just mentioned, is if two organisms are able to mate and make an offspring that is capable of mating, then we would say those two organisms are the same species, right?
Yeah.
That's what you're getting at. Whereas sometimes, like, what is it, the donkey?
Yeah, donkey and horse, they make a mule, but
right.
Mules can't reproduce.
Right. So in that sense, we say that the donkey and the horse, they're two separate species, right? Yes, they are able to make an offspring, but the offspring is not able to reproduce. Correct. So in terms of, I don't know about like where the origin of lemons, was it lemons or limes? It was lemons, right?
Yeah, lemons.
Okay, so I don't know the history of the breeding strategies that occurred in order to get lemons. I can learn more. I could look that up and tell you more about it. I will say though that often in things like citrus or in apples or any food product that
Yeah,
we eat.
because I know in like plants, there are a lot of hybrid species. Like I think
Yeah,
what's the other one? Pomelos.
yeah, it's like, it's weird though, because you have to think about it, like often those are the results, right? So yeah, sometimes to make different food products, we will make two separate species. And the result could we might use pollen from one species to fertilize another species. Plants are a little, like, when it comes to biological species concept, plants are a little weird, because you do have instances of hybridization quite a lot in plants. It's actually a pretty common thing in plants. But my point is that sometimes you have to think though, like in order to get a pomelo, like, or an orange or a tangerine or a lime or a lemon, evolutionarily speaking, they're all part of the same lineage that led to like citrus. And so they might have originally been a single species that now has either split over evolutionary time to be different species, or as you said, hybrids.
Okay.
I don't know if I answered any of your questions. The other cool thing though that I keep thinking of is, so you know navel oranges, like oranges that have no seeds in them?
Okay.
I don't know if you ever seen it. So like they're called navel oranges. You can get them a lot at the grocery store. So navel oranges are seedless, and they came about because there was an orange grove in California that there was a tree on that grove that had some mutation. Maybe it was a hybrid, and it never produced fruit that had seeds in it. The fruits were always seedless. And so every navel orange that we've ever eaten actually has come from that
One tree.
Yeah, so they grafted it like so many times. And ironically, that grove that tree was in has since been destroyed, and there's like highways there now, but that tree is protected.
Really?
Yeah.
I never know about that.
Yeah. So yeah. Interesting question though. Thanks for making me think about that.
Thank you.
Anyone else have any questions? All right, everyone. Well, if you think of anything, feel free to email me. Have a great day.
I have one more question,
Oh.
Professor. Sorry.
Okay, Andrew.
Sorry about that. So after the assignment is like after the due date passes, the assignment isn't going to show up under the assignments section in Blackboard. Okay. All right.
Yeah,
Just
so
double check.
what you have to do is you have to email me. If you miss the due date, email me and I will reactivate the assignment for you. But you only have 48 hours to do that after the due date. So keep that in mind.
Okay.
All right.
Because I missed this one too.
Okay, so send me an email so that I can reopen it for you.
Okay. Thank you.
Yep.
Bye. Have a good day.
You too. Bye, everyone.